Hey, did you know that bioinformatics can contribute to climate solution and environmental sustainability?
Oh, I'm not quite familiar with applications of bioinformatics.
Okay, tell me, what comes to your mind when you think of bioinformatics?
Things like genomics, transcriptomics, metabolomics, and metagenomics.
Bingo, metagenomic. That's the word I'm looking for. This is how scientists use bioinformatics to give insight on climate and environmental sustainability.
Oh, do tell me more about it.
So, to begin with, when we are talking about collecting DNA, what comes to your mind?
I'm not really sure. If I'm not mistaken, we collect the sample organism, we homogenize the sample, then we collect the DNA.
Yes, indeed, that is correct. This type of sampling is called invasive sampling. The organism in question have to be captured for this to work. But what if I told you we could detect the presence of that organism even without catching it?
Oh, that sounds interesting. How is it conducted?
You see, each organism shed their DNA to the environment. Cells, secretion, waste, gametes, and decaying matter all contains the DNA of that organism. Hence, we can use this environmental DNA to detect the identity of that particular organism.
Oh, I see. Does this mean that, for example, we have pests growing on our produce in a farm? We can tell what kind of pest it is just by analyzing the DNA of the surrounding area.
Yes, exactly. You're getting the point.
Wait, but how exactly can bioinformatics be so prominent in the contribution to climate solutions and sustaining the environment?
Well, why we use bioinformatic tools is because of a few reasons. First, like I mentioned just now, the new sequencing technology like new generation sequencing coupled with eDNA enable us to identify microbes without invasive sampling, so we won't disturb the natural environment. Second, bioinformatic tools enable us humans to design climatic solutions to solve climate issues, for example, using the tools to identify and supercharge enzyme in algae to remove atmospheric CO2.
Wow, that is amazing. Now we can get rid of excess atmospheric carbon dioxide and reduce global warming and greenhouse gases.
That's not all actually. We can also predict how organisms shift as climate changes by merging satellite with genomic diversity metrics. This way, we can tell which species are at risk of extinction and carry out corresponding action.
Now that you mention it, I saw it on the news the other day that they are using bioinformatic tools to clear out pollutants in the environment. I remember hearing about computational tools being utilized to map out microbe metabolic pathways. This allows us to understand how microbes degrade pollutants like oil, plastic, or heavy metals in the sea.
Yes, exactly. You're getting the point. Let's get Sotapa and Josh to do the explaining.
Hey Sutapa, do you know that more and more scientists are tending toward bioinformatic approaches in climate and environmental sustainability solutions?
Oh, how come? Is it the discovery of eDNA and metagenomics that allow for non-invasive sampling of the organism present, or perhaps it is the much lower cost of sequencing in the current landscape?
You are so close. Yes, the non-invasive sampling of eDNA and metagenomics, coupled with the low cost of high-throughput sequencing and the improvement of bioinformatic tools such as denoising pipelines, machine learning classifiers, allows for the rapid rise of bioinformatic applications in the field.
I see. Since eDNA can detect all the species present in the ecosystem, coupled with DNA barcoding, we can identify all the species present, right?
Yes, but with a grain of salt. You see, presence of eDNA does not necessarily mean an organism has been there. There might be a possibility of DNA drift, which we'll discuss later, but the important point is that we need to interpret the results very carefully so that we don't reach an incorrect conclusion.
Speaking about barcoding, I think I've heard of it somewhere. Isn't barcoding just assigning a particular genetic sequence from a species so that other people can detect the organism later by referencing the DNA barcode?
You are absolutely spot on. There are several genes chosen as barcodes for different types of organisms. For example, for animals, the COX-1 gene was used, and for bacteria, the 16S rRNA gene was assigned. While for fungi, the internal transcript spacer or ITS gene was assigned. Multiple gene loci can also be used to increase the accuracy of barcoding for a type of organism.
I see. So after we sample and sequence the eDNA, now we can run the sequence to a database of DNA barcodes to determine the species present in the ecosystem.
Yes, using this method, we can target, for example, a pest that is infecting a forest tree, using its eDNA to confirm the identity of the pest. Then, we could administer a proper treatment targeted towards the pest instead of using a broad spectrum pesticide.
Wow, that's crazy. Wait, having the ability to search the sequence with a DNA barcode database means we could identify multiple species present in a complex environment, such as the entire soil microbiota, correct?
Yes, indeed. In fact, scientists have been using this approach to compare changes in microbiota in treatments mimicking climate change to see the effects of it on microbiota populations.
Wow, that's genius.
Yes, genius indeed it is. According to scientists, there are two main ways to perform metagenomic studies. It's called the targeted metagenomics and the shotgun metagenomics.
Shotgun, that's such a cool name. I assume shotgun metagenomics is sampling everything under the sun, while the targeted metagenomics focuses on one part of the gene.
Yes, but not everything under the sun. So in shotgun metagenomics, scientists will break the eDNA into small fragments and sequence everything using an NGS to create a sequence library. This is useful when identifying particular genes such as antimicrobial resistant genes present in the environment. But the processing of this method is much more complex and limited by the sequencing depth during next-gen sequencing.
Oh, and let me guess, the DNA barcode loci is used for targeted metagenomics.
Yes, you are right.
So, targeted metagenomics, also known as amplicon sequencing, which focuses on using PCR primers to amplify a targeted region of a gene from an eDNA sample, such as 16S RNA gene, to gain information about the microbiota present in the ecosystem.
So with this, we can know the taxonomic diversity in that ecosystem.
Yep, that's correct. Let's look at a more concrete example, shall we?
Oh hey look, there are a few scientists over there performing a study. Let's have a closer look.
Hello, kids. So what brings you here today?
Oh, we're looking around for examples of applications in bioinformatics regarding climate and environmental sustainability, and we were led here.
Oh, yes, you have come to the right place.
Would you mind sharing with us about your research?
Yes, dear, of course. We noticed that many traits are phylogenetically linked between species.
And so, we're looking to create a framework to predict microbial responses to global change.
Oh, I noticed there is a sign for response trait and functional trait. What are those?
Response traits are traits that determine the microbial abundance in response to global change, while functional traits are those that affect ecosystem processes such as nutrient cycling.
So, are you saying that the response traits and the functional traits are phylogenetically linked in the related species, and by monitoring the change in that certain species, we might get an idea on how other related species might perform in the same changing climate conditions?
Yes, dear, that is correct. Hence, we only need the response of a few selected groups with documented response to predict the changes in abundance and how it affects nutrient cycling during a changing climate.
Oh wow, brilliant. Might there be any challenges that is faced in this particular field?
Oh, sure there are. The current database for environmental metagenomics is still far from completion and will cause bias towards reported organisms. We cannot detect an organism we don't know yet.
Environmental variability may not be considered due to insufficient data throughout several years, as changing seasons may also cause changes in microbiota besides from the changing climate.
I heard that there's also a risk of eDNA drifting into sampling site, causing false positive result and false reporting of organism.
Yes, dear, that is absolutely true.
From the looks of it, we need another project like the Human Genome Project to create a complete database for organism referencing.
In fact, there is. It is called the Earth Biogenome Project.
And we need to standardize the field sampling and lab workflow to ensure each team is working with the same protocol.
Oh, and shouldn't eDNA studies be continuous rather than one-off studies? So that there will be constant monitoring and data about changes in the environment in real time.
That is true. I actually hadn't thought about that. Great idea, kid. I'll pull some strings from the United Nations and write a grant proposal for it. Thanks.
Thanks for the sharing session.
Thank you, my dear scientists.
You're welcome, my dear.
Hope to see you soon working in this field.